Hey, Dr. Goodenowe.
Hi, Victoria.
I loved that lecture that you just gave on the Neuro MRI technology you've been developing.
It's really exciting. I think what's interesting about MRI is that people think of it like a picture. It's an image, but it's so much more than that. It actually probes into the structure and function of the human brain. And so it's pretty exciting once you start, peeling back the layers of the onion, if you will.
Absolutely. And it reminds me of your ion cyclotron technology and what you've been able to do with blood work in assessing somebody's health and their biochemistry and the prodromes of their health and the way their health, the direction their health is going, right? And now
Exactly.
you've kind of brought that same mindset and are now digitizing the human brain.
I know. It's really interesting because when I invented the ion cyclotron non-target metabolomics technology, right, back in 1999, 2000, it was caused by the same type of unmet need in the marketplace, right? As a scientist, we're trying to understand the biochemistry of human cells and living systems, and we had no way of comprehensively looking at the biochemistry. And the ion cyclotron technology was capable, if I was able to develop the right methods and tools and programs to actually measure human health at that comprehensive level. And what's interesting is that the ion cyclotron uses a superconducting magnet. So we actually use these 7 Tesla magnets here at the facility for our metabolomics analysis, which is the bioscan. And so MRI, of course, also runs on superconducting magnets, which is kind of an interesting thing. They run a 3 Tesla, which is still, it's a big magnet because it's got a wider bore for a human body to get into it versus, a mass spectrometer. But the same kind of concept is that we have these tools that have been around. People measure, they think, we're going to go get a brain MRI, right? If you go for hospital, you have a stroke or something. But most people look at the MRI and think, it's kind of like a picture of my brain. But it's really not. It's actually, they can use it to make a picture, but really that's not what it is. It's measuring how water moves in the brain, and then that can tell us so much information. You can use it just to image the brain, but how the structure and the function is really designed. And the real challenge has been that advanced MRI technology, there's so many scientists and research facilities that have been advancing MRI tools over the last 30, 40 years. But virtually none of those tools are available to the general public or even to our advanced practitioners. And so we have this technology, but no one really has access to it.
Yeah.
And how do you create a package? Because sometimes it takes a long time to get these scans. So part of it's designing something that you can do within a reasonable period of time. So this Neuro MRI, so 45 minutes to an hour, roughly. So still, you're in the scanner for a long time, but there are several, there's 9 or 10 different sequences that we run.
Wow.
Each sequence measures a different part of your brain. And then that all gets put together again. And what really started this for me is that I wanted to know more about brain physiology, brain structure, aging of the brain. And I had researched all these papers, and we do all this, work with advanced doctors. But even myself, with my connections, getting a full comprehensive analysis of my own brain really wasn't even possible. 2 or 3 years ago, I wanted to do this, and it can be, the sequence can be done, but there's really no one around that can actually quantify, digitize it, get it into a format that can be done at scale, for everybody. And that's really what the last 3 years of developing this Neuro MRI platform has been about, is really getting it to a point of robustness and reproducibility and completeness that we can now distribute it through all of our doctor network now. So all of our doctors can have access to
Yeah,
this
that's
technology.
no easy feat to package all of that and put that together. And I think some people maybe have heard of volumetrics or NeuroQuant, or the different programs where they can look at the brain volumes as kind of what's on the cutting edge of brain imaging. And in your lecture, I found it so interesting that you said that volumetrics can be useless and even misleading.
Yeah, it's so this is really what is a dangerous situation. not just in MRI, but really in many, many other health technologies, is that we do these large population studies. And as a large group of individuals, you'll see trends and you'll see statistical associations. And those are true. Those are statistically valid things that we measure scientifically. But applying population data to an individual is always very, very dangerous because everyone's got a different brain. Your skull size is different. The different lobes of your brain will have different volumes. And so there's no one human brain. Okay? Each brain has a number of unique characteristics. There's obviously generalities, right? We all have the same, we all have a hippocampus. We all have a frontal cortex, right? But when you look at your hippocampus and you say, is this a good size hippocampus? Is this my natural size? The question really is how has your hippocampus changed? And can you maintain its health over its period of time? And volumetrics can be very dangerous because your brain volumes will change based upon your physical activity, based upon whatever toxins you may be exposed to or other stressors in your life, and so on. And for example, inflammation can cause changes in brain swelling. Children with autism, okay, when they're born or early, one of the characteristics of an inflamed brain in childhood is increased head circumference. So just having a high volume is not necessarily a good thing. So volumetrics, as a general tool, very valuable. Like, your ventricle volumes should not be growing. Like that's pretty straightforward. And your overall brain volume and shrinkage shouldn't be shrinking. That's absolutely true. But when you get into the individual regions of the brain, it's very easy to misinterpret data. And that's when you're trying to deal with, what we're trying to do is really understand the aging process and reversing the aging process, not just by guesswork, but by systematic, detailed, targeted actions. Okay, how do we actually, test, do an intervention and measure the effect of that intervention? Then it turns into a whole different ballgame now. So we need to understand exactly what's going on.
Yeah.
And that's just not available with volumetrics or other basic
it seems like it's a piece of the puzzle, but just that. There's a lot of other things, and we'll get into this later, but I think your own brain, you showed that your hippocampus volume actually went down, but your brain function improved and your memory actually improved. So we'll get into that a little bit. But so beyond volumes, the four main things that you spoke about with this Neuro MRI are cortical thickness, neurite density, volume, and then connectivity.
Correct.
Can you talk a little bit about each of those and why they're important?
Yeah, it's really kind of interesting because you think of the brain and people, the brain is really a mystery to most people, right? You see a picture here and you see a picture there, but as you dig into it, it can become complicated, but it's actually not that complicated. Like it can be understandable by everybody. But yeah, so the overall volumes that you mentioned are important because your brain shouldn't shrink, first of all, okay, for sure. Then the cortical thickness is that band. Like everyone sees a picture of a brain and you see how, all the folds of your brain, that outer cortical surface area that they call it, right? Well, that's a band of cells. Your gray matter is a band, and there's a thickness to that band. But that's not just some generic amorphous group of cell bodies like neurons. No, there's actually 6 layers in that cortical band, right? And so, for example, when you feel something on your hands, that is going to be what's called an afferent fiber. That'll go to your cortex region, but it goes to one of the layers, and then it communicates within that layer, those 6 layers, and then it'll go to what's called an efferent fiber, which is an outward fiber, to tell you to move your hand, for example, right? And so that cortical thickness, those 6 layers, are interacting with each other. And the more interactive it is, the thicker it becomes because it's like building a muscle, right? So as that cortical thickness is exercising, it's like your muscles grow because they're being used. And that the thickness of the cortical band is really highly related to your function. Not just, so your motor function, your emotional function, your cognitive function. So all of our activities that we think of are all dealt with in that band of cells, and it's a lot of the interactions happening within that band.
And that you showed on your own brain.
Correct.
Improved dramatically.
Dramatically, yeah.
Right? The thickness, the cortical thickness, and there's this area kind of that looked like if you were wearing a headband it was dark blue, which meant very thin.
Yeah.
And then your repeat scan a couple years later, the color was dramatic. I mean,
Dramatic,
it was
yeah.
undeniably different. That's remarkable that you can actually change the thickness of your brain.
Yeah, so the increase in my thickness went from, I think it was like from 2.38 to 2.41, 42-ish, and every measurements will have, every technology or every setup, there'll be slight differences from lab to lab. But that's about a 15 to 20 year reversal. It normally takes, for my brain to, if it was in reverse, if my brain went from a 2.42 or whatever to 2.38, that would normally take about 20 years to shrink that much, right?
Wow.
15 years or so. So that's basically a 15-year age reversal from the normal rate of decline.
And how is that thickness different than the general volumetrics?
So the volumetrics kind of takes larger regions, and it kind of, like thickness and volume does correlate to some degree, but the thickness is really about that ribbon. And you can have, it's similar but different.
Yeah.
The thickness is much more responsive to changes in function of the human body. So
and it's that ribbon shape that we think of when we look at the brain, and that's the gray matter thickness that's on the outside.
Correct. And we parcel it into 600 regions, right? So even for our advanced volumetrics that we do, like we measured 253 brain regions, but those aren't 203 unique. It's basically around, around 200 good regions that you can measure. And cortical regions, you're less, maybe 150. So the resolution of cortical thickness is much higher. So instead of just measuring 150 regions, we're measuring 600. And so you're getting a much higher resolution of the cortical map.
Interesting.
And those things correspond to specific physiological functions of the human body.
So we talked volume, cortical thickness, and then neurite density. So this is how densely the neurites are packed in. And that was was it that your hippocampus was more dense?
Yeah, see, that's where this called this neurite orientation and dispersion and density index, right? This NODI value. It really is a game changer in understanding the structure and function of the brain. It's an advanced version of diffusion tensor imaging. Maybe people have heard about DTI, for example. But NODI is extremely computer intense processing. But it's really a game changer when it comes to understanding brain health, because what it does is it takes every single voxel or three-dimensional cube of the brain, what the MRI kind of will isolate for you, and it looks at what the water is doing in that region. And it defines the water into one of three areas. Either it's in an axon or dendrite. So you look at the wiring of your walls, like all these long fibers that have an axon and a protective coating, so there's myelination that sends signals long distances or short distances as well. But it's the connectivity. So the axons and dendrites is the connections in your brain. That is measured, called intraneurite water. And then it also then says, okay, because that water has a very unique characteristic. It moves in a very specific way. And that we can identify which water is moving in that way. And then there's another type of cell water called your cell body. It's like a sphere, for example. So the intraneurite water is like water that's constrained into some sort of cylinder, for example, like a wire in your wall. Whereas the extraneurite water measures water that's in a cell. So rather than moving in a linear direction, it can move in a sphere, but it's still constrained. And that's called, water that's in a cell body, like a microglial cell or a neuron cell. And then the third part is free water, water that's not constrained at all. It's just like floating, like water in my cup, for example, right? And that's really powerful because that tells us a lot about what type of cells are in that particular cube. Okay, how many cells are in that cube? So if your brain is inflamed, like if you get sick with an inflammation of the brain, the brain's going to react. You're going to get microglial inflammation. So, when people say inflammation in the brain, that means your brain immune cells have proliferated. You got a whole bunch of new immune cells. So inflammation is a whole bunch of these inflammatory cells. So all of a sudden, now you have a whole bunch of more water being constrained in those cell bodies. So now nobody can show, if there's an inflamed brain, that there's an increase of this called extracellular, extraneuronal
So
water.
the volume could be bigger
Exactly.
because of inflammation, or it could
Yep.
be bigger because it's just really dense and big. And
Correct.
you don't know until you look at the different breakdown of the
Correct.
where the water is.
And the cells that really matter in terms of actually quality of the brain is this intraneurite water. As your brain is healthy, the more connections it's making, the more healthy those connections are. That's all reflected in the intraneurite water. Okay, so intraneurite water is the number one. The water that's in those little cylinders, that is the number one characteristic of a healthy brain.
Wow.
Okay, so if you have multiple sclerosis, for example, and you have an inflamed brain, and you start seeing demyelination, what happens is that intraneurite water goes down, and your extraneurite goes up because you get more cell bodies. So of all the variables, this intraneurite water is super critical, and it's so easy to interpret. It's unambiguous.
Yeah.
It's how many good fibers you have in your brain. And so in my situation, where you saw, like, from a gross volumetric perspective, generally speaking, my volumes were increasing. My cortical volumes were increasing. The white matter was increasing. I mean, the gray matter was increasing quite nicely. And what was anomalous was that my white matter seemed to decrease. And white matter is really susceptible to inflammation. So what you don't want to happen is you don't want your gray matter and your white matter both to go down. Okay, that's definitely a bad thing. So those are, but if one is going up, if your gray matter is going up and your white matter is going down, that means your brain's getting healthier in many ways.
What's so interesting is, like, how would you ever know these things?
And so for me, what's interesting, serendipitously, right? And it wasn't until after I did all this MRI that I realized I had to go back in time and say, well, I really was suffering from post-concussive syndrome for 45 years. Like, I got a really bad head injury when I was around 7 years old, or 8 years old. And I talk about this in these concussion lectures about traumatic brain injury and how long it lasts. It can stay in your brain forever. And my brain is a testament to that. But then it also gives me the directionality in terms of how do we actually go into the brain and rebuild it and restore it and reverse that inflammation and reverse the aging of the brain. So DOTI is very important. So this ability to take at a voxel to voxel, at a cube by cube level, that what is the quality of that piece of brain tissue? Is it inflamed? Does it got good axons?
Not just is it big or small.
No, it doesn't matter. Well, because the size is irrelevant. The voxel is a voxel, right? It's defined by the MRI. So it's not. There's no such thing as a big and small voxel. It is what it is. But you can say, okay, what's in that? What's in that cube? And you say, okay, is it got a good density?
What's the composition
What's
of
the
the
composition
cube?
of it, right?
That's so interesting.
And that is independent of volume. Completely has nothing to do with volume whatsoever.
And then there's many things you're measuring, but if we're talking about core components, we talked volume, cortical thickness, density, and then the last one is connectivity. So how the brain kind of talks to itself. How do you measure connectivity?
Well, this is really exciting. So people get a little distracted with this we call functional MRI, which is still interesting but functional MRI is active. Like, our functional MRI between you and I right now is changing as we speak. Okay, functional is a
Snapshot in time, right?
Yeah, well, the technical term is BOLD. It's called blood oxygenation level dependent imaging, which means as your brain cells fire, they use oxygen. And so if brain cells in my frontal cortex and my occipital cortex are firing at the same rate, it shows that they're connecting functionally. But the question is, how do they connect functionally? Well, they connect functionally through a physical wire. There has to be a physical wire that connects my frontal cortex to my occipital cortex, right? And that physical wire can be measured. It doesn't change whether you're sleeping or awake or whatnot. It's a physical connectivity. And that's where this diffusion tensor imaging connectome comes into place. And so we can now take this advanced technology and we can measure all these fibers. And the MRI will measure somewhere between 300,000 to 600,000 fibers, okay? All these little fibers. And so, because sometimes neurons are connected with very, very short distances, like within a millimeter, and sometimes the connections are very, very long. And so what this tractography does is it measures all of these little, strings, if you will, And then it combines them all, these 300 to 600,000 ones, to one region and the other. So we can say, okay, what is the connection between your frontal cortex and your spinal cord, for example, or your brainstem? And that tells you the total amount of actual myelinated fibers in your brain. And that is probably the most important measure of true structural health of the brain, because it tells you it's the wiring. How good is the wiring of your brain? And this is that fourth component of understanding general brain structure and function.
It's like
pretty cool.
If an MRI was, taking a snapshot of your house, volumetrics might tell you like size or square footage, but now we're looking at, is your electricity working? How's your plumbing working? How's your AC? Like, it's really looking at the actual component parts of what makes it, function.
Exactly right. Yeah, it's the connectome basically is the hallways. How do you get from one room to the other? Okay, the DTI is measuring. Okay, are you a hoarder? Do you have a is your room?
What's in that square foot?
What's in that bedroom? Right? It should have a bed and a sofa, and a dresser and so on and so forth. But if it's full of a whole bunch of extra boxes, you're gonna say, oh my goodness, something's wrong with this bedroom here. This is so. Now that you think about it, I don't want to do a noti of my house. but you have that extra room where you put everything.
Everything,
in. Right.
all of the junk.
But that's basically what it's doing.
It's so
really looking
fascinating.
at it.
So tell me, if someone is healthy, should they still get one?
Well, that's the most important part, because you need to know your brain. Your brain is you. And also, I can't tell you how many times people come to me after they have a car accident or they have something happen to them and say, show me what, something's not right. I've hurt myself. Show me what's wrong with my brain. And they think that all these technologies are perfect, and they're not. Okay, like they're trying to diagnose a concussion just out of the blue. Okay, is difficult. Like there'll be telltales, but your own human variability is quite high. It's very important to get your own baseline as early as possible, especially when you're feeling healthy. Okay,
in fact, that's a good time to go, right? Because then if something changes, and it's not about a diagnosis, but you just know that something is different, you can look at what your brain looked like when you were feeling good and when you were functioning well and have that as a reference point.
Exactly.
It's less, and I love what you said about the population database, because yes, those things are probably important for large scale connections and, everyone has certain parts of their brain. But what really matters is you compared to you, right?
Yeah. And everything we talk about with our prodrome technology and understanding, we're so focused on diagnosing disease. Okay, and MRI, a huge amount of the MRI technology has been developed to say, can I use MRI to diagnose Alzheimer's or Parkinson's? And what we do differently with this whole perpetual health model that we've created is saying, no, we're going to ignore disease. Like disease is something that happens way after a deviation from health occurs. Like the brain is supposed to be structured and function a certain way. And we can use the MRI to understand that. So when you're young and historically speaking, the human brain seems to reach its maximum functional capability in your 40s and 50s typically, because your brain continues to myelinate. As you learn new skills and new things, your brain is getting better and better at doing things. And it seems to have this progressive improvement or optimization up until our 40s and early 50s sometimes. And then the wheels fall off basically, and the brain starts declining. And this study's been shown over and over and over again. So really, what you want to know, and what we're trying to achieve now, with all of our work with the bioscan and the blood work and the NeuroRestore and the targeted supplementation protocols, is how do we basically prevent aging? How do we maintain perpetual health? But so maintaining perpetual health at the brain level is not having any brain shrinkage, improving your neurite density, and it should reach an optimal plateau, and you want to keep it there, right? And so even what we now know historically in that 40 to 50 range, well, it could be better than that, right? So now that we know how to actually improve neurite density, how we actually know how to reduce inflammation in the brain, how we know to improve connectivity metrics, so these historical data, we can start changing that and creating optimal brain health. And if by doing that, you can track your own optimal brain health. And once you get to that optimal state, you can keep it there. And our goal, obviously, is to maintain that till you're 200 years old, basically.
Yeah, I think this technology is so amazing for many reasons, but I think your stories, the Lisa Grillo stories of re-showing, like growing different parts of the brain, connecting different parts of the brain, basically the fact that we can improve health, right? Like the technology is amazing, but a lot of people probably wouldn't want to know if there is nothing to be done.
Exactly.
Why would you want to know that your brain is,
And we're really changing the thought process here because the human brain is incredibly dynamic and it is incredibly regenerative, much more than people have ever given it credit for.
It is able to change.
And like the changes in my brain are really out of this world.
And are they reflected in your experience? Like,
yes.
do you feel cognitively more with it?
Oh, absolutely. And, my wife will tell me, like, I remember trivia questions better than I've ever have in my life. But the issue, like for me, like from a real structural perspective, when you look at the cortical thickness regions, two main regions that improved were my visual, my central visual and peripheral vision. And absolutely, okay, my vision has basically been restored to when I was 17. Okay, like I don't get headaches. I hardly need my glasses anymore, except for even when I was 17, I still had it. I was nearsighted, so I still used glasses for playing hockey and long distance stuff. And so but that's kind of where I'm at right now. So absolutely, my visual performance has completely restored to normality. My neuromuscular performance has improved. So yeah, the physiological correlates of what I'm seeing on the brain scan and what I'm feeling in my everyday life are absolutely correlated with each other.
What are you excited about the most with this technology?
Well, what I'm excited is giving people hope, and we can actually take an individual, like take Lisa Grillo, who was blind for 30 years, had no vision in her right eye for over 30 years, and she can now read out of her blind eye. Okay, that is pretty crazy, and so that means what we've considered lost causes of the human brain are no longer lost causes. And we can track that performance improvement in the brain. And we have targeted nutrition that can get into the brain, that can restore mitochondrial function, restore membrane structure and function, and we can see the brain grow. Now, you need to marry that with good lifestyle modifications, right? Like you need to actually use your brain and use your body. Like Lisa Grillo is improving because she's out there living her life and she's active and she's doing things. So the human body still has to be functionally active. You have to have purpose in this world and so on and so forth.
And another major component of that was your NeuroRestore protocol, right? That's what is kind of the foundational thing that's helping with the neurological changes and increasing cortical thickness and things like that.
Without NeuroRestore, you're really just doing random acts. Like there are certain things we know do help brain health overall, like resistance training for exercise, B vitamins. Like these things have all been shown at different levels to have modest improvements, but nothing has ever been able to have real dramatic changes and sustainable improvements and maintenance over time. And that's what NeuroRestore is about. Like it's designed specifically for brain biochemistry and health. And without NeuroRestore, it really doesn't make a whole lot of sense. All you're doing is morbidly tracking your death, fundamentally. Okay, and people, or you're just guessing, right? You're basically saying, everyone says, eat well and exercise. Well, that's true to an extent, okay? But there's lots of healthy people out there, physically healthy people who take star athletes, for example, right? So some of our best athletes in the world end up with, they've had too many concussions so there's many other things that will negatively affect you. So now we're at the point where we can actually measure, intervene, and track that performance outcome. And we can measure it at the blood level, and we can measure it at the brain level. And between those two things, we can really achieve this goal, which is perpetual health. Like we actually can do it now. It's pretty exciting.
I think really the brain has been such a black box for so long, and it's going to be amazing to see what we find now that we can actually look inside and see how it's operating.
Oh, it's pretty exciting.
Absolutely.
And we can put all this data together now as well. And so there's a lot of, we're just beginning, but at least what we have now, we have really a core, foundational, comprehensive model of the brain. Like we can digitize the human brain, and there's no escaping the Neuro MRI. Like if there's something wrong with your brain or good with your brain, it's going to see it and you can move it. So despite all of our focus on the health and optimizing health, it still does all the things, you know, an important neurologist would want for brain health. Like it still looks for brain bleeds, it still looks for lesions, and all those other things that a pathological neurologist would want to look for. They're still there as well. Okay, so you're still getting that component, like what you would normally get at an emergency room MRI. Like it still does standard MRI stuff for people, and they can send this information to whoever they want to review it. But for the advanced perpetual health work, where we're saying, okay, how do we restore the health of the brain? That's a space that the neurologists of the world just aren't involved in. And this is a whole new world of building the brain and tracking its strength over time.
And combining that with the blood work and all the other technology, it's just going to be amazing, the things we find. I hope we find the new plasmalogen, and we keep going.
Keep going. Well, it's one thing, we just keep, it really, well, we actually have this muscle water. We'll talk about that later on. It's been, so part of our clients with muscle wasting disease has been a big challenge, or you get these hypermetabolic states, and people can't maintain their weight, they can't maintain muscle weight. And so I've been focusing a lot on neurological health and membrane structure and function and health, because it's a critical component, right? Parkinson's, Alzheimer's, MS. But we've designed a muscle water that's designed to maintain muscle fitness, and it's quite dramatic. It's really dramatic. Like even myself, like literally in using this, keep my exercise routine normal, I have basically converted 6 pounds of fat into 6 pounds of muscle in 3 weeks. Not changing.
Using biochemistry?
Yeah, it's incredible. So stay tuned. It's called My2O. We'll have to do a talk on My2O.
Okay, we'll do that one next.
Yeah, it's pretty exciting.
Awesome. Well, thank you so much again. This was awesome, and I can't wait for the next one now. Got to hear more about that.
Got it. Okay, bye.
Thanks.